Okay, so let's talk about the effector functions of these various antibodies. In other words, how does the humoral immunity ultimately function and how does it fit in with other branches of our immune system? And I'm actually going to skip over these next two slides. This one, whoop, never works. There we go. This one and this one because I'll talk about these. I mean, you have them in front of you, but I really want to use this picture because this summarizes all of the various aspects of how the end functions of these antibodies work. Remember that our antibody, the FAB portion, is going to bind to the epitope. So this could be an epitope on the surface of a bacteria or maybe a virus or some pathogen, right? And so the FC portion is sticking out into the external environment. And this is basically like a big red flag, right? This is the signal that's going to determine how the rest of our immune system sees it and therefore what downstream effects this antibody will have. So I've always thought of, I feel like there's a lot of overlap between antibodies and complement. And I've always had this kind of idea that I've never really looked into too much, but I feel like antibody is kind of a more evolved, I'm not using the word evolve right, I shouldn't use the word evolve, a more advanced corollary to complement. They have a lot of overlapping functions, but remember that complement is only in multicellular eukaryotes, and antibody is only in, complement is in all multicellular eukaryotes, whereas antibody is only in chordates, right? Non-chordates don't have adaptive immunity. So I just find it interesting that they're both secreted proteins, right? They're both really small. They're not cells, and they both have kind of a fixed activation. How should I say this? A fixed structure that determines their activation and what they actually do. And they both have similar downstream effects, but they also have overlapping. They also work together, right? Like antibody can activate complement. I'm going off on a tangent. Let's focus, Sarah. Okay. So what does antibody actually do? Well, here's one of the ways, for example, that it acts a lot like complement. Remember that complement, I think it's C3b, can bind to bacteria and basically overcome the capsule that a bacteria can have, right? And that complement can recruit phagocytes. And not only do they help recruit phagocytes, but then it helps that phagocyte grab onto the bacteria and opsonize it eventually for digestion and destruction. And so it turns out that antibodies do the exact same thing. So their FC portion can bind to that phagocyte, and it makes it super easy for that phagocyte to gobble up that bacteria. So it can do that in the case of a single bacteria or a single virus right here, but it can also do it through cross-linking. So it can bind to many bacteria at the same time, kind of forming this cross-linked chain. And what that does is it not only immobilizes the bacteria and gets them to kind of stick together, but now this phagocyte can gobble up the whole thing at once, making these phagocytes much more effective in terms of clearing that infection. So in a similar fashion to cross-linking, basically this immobilization, it can do that on its own. So it's not just an phagocytic function through cross-linking, but it can basically bind to, in this case, it's binding to the flagella of this bacteria, and that's not allowing the flagella to move. And of course, if a bacteria is modal, making it non-modal is really going to hurt its chances. Remember that they also have pili. They have these little external extensions that help them adhere to things. So IgA, for example, is really good at this. They're really good at binding to flagella and pili and basically external structures of cells to prevent them from adhering to our epithelial cells, for example. And they basically form these sticky traps in our mucus and immobilize these bacteria. Kind of an extension of this to me is neutralization. So similar idea here. They're just simply going to bind to whatever that pathogen might be. In the case of a virus, if that virus is bound to an antibody, that virus is now not free to bind to a cell. And of course, as you remember, the only way that a virus can survive is getting into a cell. So if a virus stays outside of a cell for too long, it's just simply going to die. So if this antibody is able to stick to it, it's going to prevent attachment to another cell and thus end up killing the virus itself. We haven't really talked about toxins yet, but bacteria can secrete toxins that are, of course, extremely toxic to us. For example, the botulinum toxin or the tetanus toxin are two big exotoxins that bacteria can secrete. And it turns out that if we get vaccinated against tetanus, for example, and so we can actually, that's not how that vaccination works, but we can get a antibody. One way, how am I trying to say this? One way to neutralize the tetanus toxin is through injection of an antibody that's specific for that toxin. And so if the antibody is bound to that toxin can now cannot bind to cells to exert whatever toxic effect it has. Oh, let's see, what's next? Actually, the next two, they're kind of grouped together too. So ultimately, antibodies can also direct the direct killing it's called cytolysis. Basically, cytolysis means breaking open of the cell. And this can happen either through the complement activation. So the Fc portion recruits complement. And then that will not only promote inflammation, but it'll also recruit the MAC attack. Remember, all of that complex can form on the outside of the bacteria, and that basically breaks a hole in that bacteria and causes the death of that bacteria. On the other hand, it can work through natural killer cells. So this is called ADCC, or antibody-dependent cellular cytotoxicity. Remember that Fc portion acts like a big red flag of recruitment for those NK cells. Once the NK cell is recruited to that cell, it's going to directly attack that cell with perforins and granzymes and other really kind of caustic chemicals that eventually lead to the death of that cell. And we want this cell to die because, as you can see, it has a virus inside of it. We don't want to get that. We don't want to give that virus an opportunity to multiply and break out of that cell and infect more cells. So lots of ways that antibodies have downstream effects, and notice that it is overlapping with other branches of our immune system. For example, NK cells and macrophages of our innate immune system, the complement system, and so on. So we can call these, how can I say this? So we can refer to antibodies either by their heavy chain class, so IgG, IgM, IgA, etc. But we can also refer to them by what their functional ability is. So for example, an antitoxin is an antibody that directly neutralizes toxins. Agglutinins are antibodies that can clump together antigens. I think of agglutinins and precipitins as the same thing. I don't think there's not a really big difference between them. And opsonins and so on. So I might ask you, an example of how a precipitant or how a complement or an opsonin works, for example, and you'd have to describe how an antibody leads to phagocytosis of that particular bacteria. Okay, so I hinted before, so this is where I'm going to talk about clonal deletion. And I also talked about how we have potentially billions and billions of different antibodies, the potential, I should say, to make billions and billions of antibodies in our body. And we can make up to 10 to the 9, which is what? 10 to 6, 10, like a billion, trillion? What is 10 to the 9? I feel like I should know this. Let me Google it. Trillion. Scientific notation. Okay, so 10 to the 9 is a billion. So we can make a billion different antibodies. But here's the thing. Remember that an antibody is a protein, and proteins are encoded by genes. And humans have about give or take, I guess. I don't know what the latest refined study says, but we have about 25,000 genes. So how is it that if we have 25,000 genes, we can make a billion different proteins of just antibodies? Well, it turns out that we only have like really one gene that does this really, really weird thing called VDJ recombination. And so it's due to basically three things. So we already know that we have two different chains. We have heavy chains and light chains. And so the different heavy chains and light chains, whether it's, remember, gamma or kappa or lambda, the combination of those can lead to generate some diversity. But it turns out that when we actually transcribe, so as that let's just talk about B cells. T cells also do this, but as that B cell is maturing in our bone marrow, the DNA itself is going to undergo recombination. And this should kind of sort of blow your mind a little bit, because we've always been told that our genes are fixed from birth, right? We got what our mama gave us, right? We have our genes from mom, genes from dad, and they'll never change. And that's actually not true. Our B cells actually change. They will change this very specific gene about 1 billion times, because we want to have that diversity. And instead of having all of our cells basically carry around 1 billion different genes, what we do is we carry around this one really long gene that has lots and lots of exons and introns, basically, and we keep kind of mixing up that gene. And this is really strange, because, you know what, I shouldn't even call them exons and introns, because that's not true, because that happens in RNA. So I shouldn't call, that's not an accurate way of describing this, because this is happening actually at the level of DNA. So as this B cell is maturing in the bone marrow, what's going to happen is we have 3 different regions. We have a V region, and there's 40 different kind of repeats in this V region. We have a D region, where there's 25 different different repeats in the V region, and then we finally have a J region that is it's J for joining, and there's 6 different joining segments, and then we have a stream. The constant region is where the heavy chain is, because remember, the heavy chain doesn't change, right? So this red portion doesn't change. So this VDJ region is this blue region here, which is where we want it, right? This is where we want all of the diversity in our protein, because this is the site that is going to bind to that potential antibody. So these different regions, one of the 40 here, 25 here, and 6 here, and so on, are going to be randomly selected, and the DNA in between are going to be cut out, and it's going to get stitched back together. Now, when it gets stitched back together, it's going to get stitched back together in a really sloppy way, or basically a very mistake-prone way, so that lots of, again, just basically generating diversity in those kind of regions where the V and the D and the J meet. And if you think about, so between all of the different regions that can be selected, and then the sloppy way that they're kind of stitched together that is very mistake prone, we're going to generate lots and lots of diversity. Now, so the analogy that I like to give in class, like imagine that you had to make the world's best chocolate chip cookies, right? But you had no idea how to make cookies. Like you've never cooked before. You don't even know what any of it is, right? And you have endless resources at your disposal. And so basically what you start, and you have endless time, endless energy, and so on, right? So basically what you're going to start doing is you're going to throw different combinations together. It's totally random, right? And you're going to throw in a whole bunch of flour and 1 egg and an entire bag of sugar and 2 chocolate chips or something, right? Again, you have no idea what you're doing. And so if you think about what's going to happen there, if you do this 1 million times, 999 times or whatever, the 99% of the time, you're going to get absolute garbage. You're going to get this really nasty stuff that doesn't work at all. And that's actually what happens with B cells also. Like most of the time, when these V, D, and J segments kind of recombine, we're going to get massive mistakes, and they're going to not even be functional. And so I don't know, I'm trying to think if there's even like a percent, like if they know how many, if scientists know like how many B cells are actually deleted in the bone marrow, but it's a good chunk of them. And so this is that clonal deletion that I was telling you about. So if you don't make a functional VDJ region, and you don't make a functional B cell receptor that leads to antibodies, you're going to get deleted in the bone marrow. You're never going to make it out of the bone marrow, because we don't want garbage going out into our body. The other big deletion event is if this B cell receptor recognizes self. Because remember, we're just throwing genes together. We're throwing, or gene segments, I should say, together, and we have no idea what it's going to lead to. So just as likely as not, it's possible that we could make a B cell receptor that recognizes self antigen, which is really bad news, because if that B cell gets out into the environment, it's going to recognize self, and that's going to cause an autoimmune disease, potentially an autoimmune disease, or basically a self-reactive B cell. We don't want that. So it's a clonal deletion is a really big event in the B cell maturation process, because we essentially need to get rid of all those B cells that don't make a functional B cell receptor, or make a B cell receptor that recognizes self. Okay. I'm going to gloss over class switching because it's not terribly important. Basically, at the beginning of B cell maturation, all B cells, so we have our B cell receptor on the outside of the surface. When it recognizes antigen, it's going to get turned on to start secreting antibody. That first antibody that it secretes is IgM. And then if it's in the serum, it's going to switch. So it's going to switch its class from IgM, usually to IgG. And then if it's in the mucosa, it's going to switch IgM to IgA. All right, I got another phone call. I got to pause. I'm sorry. Okay, sorry about that. I have everything is just snowballing, and there's still 4 weeks left. Oh my goodness. Okay. All right. So class switch. Naive B cells make it out of the bone marrow, mature upon antigen stimulation, and if they're in the serum, they're going to switch from secreting IgM, which is remember that's the first antibody that's secreted, and it's going to switch. If it's in the serum, it's going to switch to IgG pretty quickly, and if it's in the mucosal lining, it's going to switch to IgA. All right, so now let's talk about MHC. This is a big chapter. Oh my goodness. So MHC stands for major histocompatibility complex, and they're proteins, and they're on the surface of our cells, and they're involved. They basically create a landing site for antigen to be presented to other immune cells. So you can see this little area it kind of creates this little pocket that foreign antigens can sit in, so our other immune cells can recognize it. And we have two different kinds of MHC. We have MHC class 1 and MHC class 2. So MHC class 1 is on all of our cells. And I kind of think of I really think of MHC class 1 as our security badge. So the badge itself, so think about, where do people wear badges? Like on the Navy base, or I don't know if they do that. What do they do on the Navy? How do you know if someone belongs there? How do you know, if Tom Cruise is just walking around? Is he really on the Navy base, or is he just doing research for Top Gun 2 that is probably postponed now, which really stinks? What was I talking about? Navy bases. Oh, MHC. Okay, hospital. Hospital's even better, right? So think about everyone that's working in a hospital. You kind of want to be able to recognize that they belong there, and so they're probably wearing a badge. And so the badge itself is the same for everyone, but the little picture on the badge is unique, and it's going to hopefully match the person, right? And that's how you know they belong. And that's kind of what I think of with MHC class 1. MHC class 1 is your security badge, and it's telling, so every one of our cells has it, and that's basically telling our immune cells are constantly surveilling and constantly looking at that security badge, and if you have it, you pass, right? So if you have MHC class 1, and in that little peptide binding site, you're showing off antigen that is self-antigen, the immune cells are going to leave you alone. But if you're showing foreign antigen or a sign of injury, then our immune system is going to take you down, okay? So let's think about MHC class 2. MHC class 2 is on professional antigen presenting cells. So APC stands for antigen presenting cells. There's only 3 cells that express MHC2, and that's B cells, macrophages, and dendritic cells. So they're going to show exogenous antigens that they have gotten from phagocytosis. So let's go into this in a little bit more detail. So let's first talk about MHC class 1. So here we have some random cell of our, it's epithelial cell or muscle cell, whatever it might be. So some cell of our body, and it has proteins. Remember, all of our cells have proteins inside. They're self-proteins. And so it's going, as part of its normal kind of life process, as these proteins kind of fall apart, they're going to get associated with MHC class 1, and they're going to be presented on the surface of this cell. And so what this is basically doing is telling our cytotoxic T cells, remember the cell-mediated responders, that, so here's our badge, right? Here, the MHC, that little brown kind of cup thing, that's our badge. And then all of these little yellow, blue, green, these are this is letting, how should I say this? It's letting our immune cell, our T cell, know that we're self, right? But if this was to present, if this peptide, or this little yellow box, right, if this little tiny protein that was sitting in MHC class 1 was, say, from a virus, or was from a bacterial pathogen, then our T cell would, our cytotoxic T cell would recognize it and then destroy this cell. So really MHC class 1, it would be like, again, if you're walking around the hospital and you have a security badge on, but that security badge has a picture of someone else, right? That T cell, the security guard, is going to approach you and they're going to say, hey, the picture on your badge doesn't match you. It doesn't match who we know you are. And so you're going to die. Hopefully not die, but you know what I mean. Okay, so on the other hand, we have these antigen presenting cells, because they're going to take exogenous antigen, so antigen from an extracellular pathogen, they're going to bring it inside through phagocytosis, right, or endocytosis. They're going to chop up this antigen and present it in the context of MHC class 2. So because they're presenting cells, they're going to present this antigen to helper T cells, because they're the ones that need to get activated. So this helper T cell is going to bump into this professional antigen presenting cell. They're going to get, if their T cell receptor recognizes this antigen, they're going to become activated and amplify those responses to the rest of the immune cells. So two very different kind of roles for MHC. MHC1 is basically helping us determine self from non-self, whereas MHC2 is helping teach our immune cells what is foreign. So here's our foreign. So our antigen presenting cell is telling this T cell, hey, this is foreign. This doesn't belong here, and this is what it looks like. So we have these billion possibilities, right? How do we not turn on all billion different antibodies at the same time? Remember, we have all these B cells and T cells. They're all made randomly. We know that they're functional because they made it past clonal deletion. And but we don't know what they can recognize. So we have all of these B cells and T cells floating around our body, really more in our secondary lymph organs, but we don't know which is the one, right? Like if we were to get infected with whatever, how do we know which is the B cell that can actually recognize it? It could be any of those billion different possibilities. So we have this really large pool of T cells and B cells that could respond. So how does that work? So what happens is the cell is actually stimulated by the antigen, or the epitope on the antigen itself. This will make a little bit more sense in the next slide, right? Okay, so here, remember that our B cells mature from our hematopoietic stem cells. And at this point, we have all of these different immature B cells, B cell W, X, Y, and Z. And we also have so again, take this times a billion, right? And that's what our lymphoid organs look like. And so it turns out that this particular B cell X, can recognize this particular antigen, this particular epitope on the antigen. So this is the one that gets the signal to go to the next step. So all the other ones, W, Y, and Z, they still hang out. They're still just kind of waiting for their match to happen. So it's only the B cell that can specifically recognize antigen, in this case antigen X, that goes on to the next step in the maturation process. And that next step is that really crucial second signal. So I've said this a couple times already, and I'll say it a couple more times, because in our immune system, especially our adaptive immune system, we need kind of this 2-signal message. And it's basically like confirmation. Do you remember, what was the name? Was it Hunt for Red October? I feel like it was called, I think it's a movie no, it's not Hunt for Red. It's with Denzel Washington and Gene Hackman. What is that? Denzel, I'm Googling Denzel Washington, Gene Hackman, Crimson Tide. Crimson Tide is a movie, and it's a really good movie. If you've never seen it, it's from like the 90s, and basically they're on this nuclear submarine, and they get like a partial signal, like comes through their comms, to launch a nuclear weapon, but the signal gets like cut off before they get the complete signal. And Gene Hackman is like the major, like the number one guy, the captain or whatever it is called in a submarine. And he's like, no, let's launch the nuke. But in order to launch the nuke, you need both the code, like you need the code from the captain and like the first lieutenant or whatever it is, like number 1 and number 2 on the ship. And number 2 is Denzel Washington. And he's like, no, we didn't get the confirmation code, so I'm not going to do it. And so of course Denzel saves the day. It turns out that it was a faulty order or whatever, and really good movie. But all that to say that if you're going to launch a nuke, like you shouldn't rely on one person, right? Like you shouldn't rely on one signal to start, like, just turn on your immune system and go nuts. And so that second signal is crucial here in order for this B cell to kind of turn into a plasma cell and start pumping out those antibodies that will then lead to, remember, complement activation, natural killer cell, opsonization, and all those things. So that second signal is given by helper T cells. The helper T cell is going to kind of bump into this B cell, and now this B cell makes a decision. The vast majority of these B cells are going to turn into plasma cells. They're going to pump out antibodies like crazy so that they can clear that infection, but a small fraction of them are not going to turn into plasma cells. They're going to turn into memory cells. And it's these memory cells that have an essential function in memory, right? So years later, months later, years later, when we see this pathogen again, antigen X, our B cells are much more ready and to respond, and can respond much more quickly. Okay, so sorry for the terrible picture. I actually replaced this picture from the one that was there, because the one that was there was terrible. It was from another book, and I didn't like it. But this one's from our book, and it's simpler, and I like it more. So remember that I just talked about how that crucial second signal, right? The T helper cell here needs to give this kind of semi-activated B cell that crucial second signal. Well, this is how it happens. So excuse me. Okay, so this B cell is also going to take in antigen. Remember, it's going to endocytose that antigen, break it into tiny little pieces, and then present this antigen on its surface in the context of MHC class 2. Remember, B cells are part of one of those three cells, macrophages, B cells, and dendritic cells that can present MHC class 2. And now this B cell is ready for that helper T cell to recognize it. So the helper T cell is going to recognize that specific antigen. So again, we not only have 1 in a billion recognizing that antigen, right? That 1 in a billion B cell. We also have that 1 in a billion T cell that's going to recognize it also. So this is why we talk about having that high specificity for our immune response, because it has to have a perfect match for a B cell and a perfect match for its T cell. And if that happens, the binding itself, so that T cell receptor itself binding to the B cell is going to give it a signal, but it's also going to give it some cytokines. So this T cell is going to pump it with some cytokines, and now this B cell is off to the races. It's going to differentiate into a plasma cell, and it's going to pump out those antibodies and help clear that infection. I think this is basically a summary of what I just talked about. ABCs, physical contact. I guess I didn't really talk about the maturation of those helper T cells. They undergo their own kind of maturation process. So there's a fruit fly hanging around me here. Okay. So they undergo clonal selection also by antigen presenting cells. And that happens here, right? So this is a naive helper T cell, and it's going to recognize that the antigen presentation, and then that T cell itself also needs a second signal, and that second signal is going to come from macrophages. So that this is kind of an abbreviation of macrophage, and that second signal comes in the form of interleukin-1, a cytokine that macrophages secrete, and it's going to stimulate the maturation of helper T cells. So most of those helper T cells are going to go on to exert their function in basically amplifying the immune response, and but some of them are going to turn into memory T cells as well. Okay, so this is a big, so the anamnestic response is a very important concept for most of you to learn because you're going into the health professions. And anamnestic response is basically the difference between your first exposure to antigen and your second exposure. So upon your first exposure, you mostly have an IgM response to that pathogen. And your antibody load will basically go away fairly quickly because IgM doesn't last very long in your bloodstream. However, upon second exposure, your memory cells are going to kick in, and your memory cells don't need that second signal, right? They've been there, done that. They've basically been waiting for this day, and so you're going to have a much faster. I'm going to skip ahead to the graph so you can kind of see this in action. So here's our primary response, and you can see that look at there's kind of a very weird x-axis on this guy. So in the time after antigen exposure, so you have this initial antigen exposure, you're going to have mostly IgM, a little bit of IgA, but it's going to go away fairly quickly again, IgM goes away really quickly, IgG will go away over the next several months. But upon second exposure, this is where those memory cells serve such a crucial function, because immediately within a day, within 2 days max, I would say, we're going to see this massive spike in IgG. And this is because those memory cells are going to go straight to IgG. They're not going to mess with IgM. And those IgG antibodies are much better at amplifying the signal, activating complement, recruiting natural killer cells, and they stick around in our bloodstream much longer. So the T-independent humoral response is an interesting avenue of study, because this is essentially what happens in people with no T helper cells, right? So people with advanced HIV, the only response that they get is IgM, because their B cells never mature because they don't get that amplifying signal from helper T cells. But it does have an effect. So they do have some kind of low level of immune response, but again, it's all IgM, and it usually happens through cross-linking of the B cell receptor on some type of from multiple cross-linking of some type of antigen. And I just wanted to point out that we don't have to have helper T cells to have a humoral immune response, but it really helps in terms of having a quality IgG-driven antibody immune response. All right, so we've spent most of this lecture talking about the humoral response, but let's kind of cross over and talk about the other one, which is the cell-mediated immune response. Remember, that's a whole other branch of adaptive immunity. We're not going to focus as much time on this as the other one because it's unfortunately much simpler. So what happens here is antigen-presenting cells, remember we have three of it's for cell-mediated, it's mostly macrophages and dendritic cells. So macrophages and dendritic cells are going to present antigen in association with class 1 MHC, and that's going to, they're going to present it to these CD8, remember that cytotoxic T cells have the CD8 marker on their cells, so cytotoxic T cells. When they're naive, this is going to activate them. And then this is going to cause them to divide and differentiate into a whole bunch of properly cytotoxic T cells. This can involve helper T cells. So you can get that second signal from T helper cells. So there's some cytokines that helper T cells secrete that can increase this response and make this response faster. But once this happens, T cells are going to essentially move on to any cell that has that particular antigen. And once they recognize that antigen, they're going to bombard that cell with perforins and granzymes. And the cell itself dies, the cytotoxic T cell survives and moves on to the next cell to kind of scan that next cell. So this is kind of what it looks like. Here we have a cell that is virally infected. So the cell is going to take some portion of that virus, right? It's going to try to chop up that virus. So again, this is kind of a timing thing, right? This can happen. So it's fascinating. One of the things that some viruses do is it actually does not allow the cell to display MHC. So it takes the MHC class 1 away from the cell, and therefore cytotoxic T cells can't recognize and bind to it. It's pretty tricky. Viruses are little devils. So here we have a cell infected with a virus, and hopefully it has time to basically chop up some piece of that virus and display it to the external environment. The cytotoxic T cell that's already been activated by macrophage is going to attach and recognize that viral little kind of peptide right there, that purple diamond that's in the middle. And once it does that, it's going to release these perforins and granzymes, and that basically punches holes into the cell. And remember, our cell membranes are basically made out of olive oil, right? They're really fragile, so it's not like a bacterial cell wall or anything. And it's going to cause lysis of this cell and basically apoptosis through damage. Another way that it can do this, another way that it can tell a cell to undergo programmed cell death or apoptosis is through ligation of a specific cell marker called CD95 and CD95 ligand. And so when those two connect with one another, it basically starts a cascading activation of proteins that causes the cell to die. So, and then I just very briefly want to mention that when MHC1 isn't on a cell, it's susceptible to natural killer cells. So even though you think, oh, that's brilliant, the virus can downregulate the MHC on a surface of a cell, now it's susceptible to the natural killer cells. Because if a natural killer cell scans a cell and it doesn't find MHC1, it's going to kill the cell via these granzymes and perforins also. So that does it for chapter 15. That was a doozy. I don't know why that one wore me out, but very complex. Adaptive immunity is, and again, if you're trying to read the chapter on this, you'll see very quickly that I'm only giving you about a third of the information in this chapter. It's a really, our immune response, very, very complex series of interacting events that kind of amplify one another and hopefully cause a correct response, right? We don't want it to respond overly. In fact, that's kind of, I think that's, there's some early evidence that that's exactly what's happening in the coronavirus response. So there's some kind of early indicators that the people that are dying from coronavirus they're basically succumbing to what we call a cytokine storm. And what that means is that our immune system basically has an inappropriately large response to an infection, and it just releases these interleukins and chemokines and complement and antibodies, and that signal just for some reason keeps amplifying, and it causes so much damage within our blood vessels, that it very quickly starts to cause damage to our kidneys. And what is the word? I think it's disseminated intravascular coagulation. Basically, we're getting hopefully not me. They're getting blood clots throughout their body, and they have very poor outcomes once that starts to happen. And it's just very interesting because it's happening at like day 10 to anywhere from like day 10 to day 17 of the infection, which is so late in the infection. Okay, that does it for now. I'm going to end chapter 15, and then I'm going to try really hard to get chapter 16, host microbe, up tomorrow so that you have a solid week and a half with all the content for this unit before the test next week. All right, I'll talk to you soon.